Thank you so much for the kind introduction. Good morning, everyone. I want to thank also Professor Barnek for the invitation and the opportunity to speak here, and also to all of you for being here. And also many thanks to all of the people that is helping around to make this a very excellent conference. So this morning, I'd like to present some of the projects going on in our lab that is focused on the application of the chemical space concept that Matthias excellently talked about yesterday, about natural products and a little bit of food chemicals. So this is kind of the outline of the talk. First, I'd like to introduce very briefly my research group at UNAM in Mexico. Then, for the sake of introduction, I'm just going to be covering the concept of chemical space, chemical multiverse, highlight the still importance of natural products in drug discovery, and then I will move to databases, putting some emphasis on the contributions from several Latin American countries. And here, I'd like to introduce like a collaborative effort between different Latin American countries to put together a unified Latin American natural product database. Towards the end, I'd like to present very briefly kind of a fun project or a side project that is kind of the merge of chemical space visualization with art. So this is going to be very brief. And at the end, I'm going to present a summary. So first things first, I'd like to thank the current members of my research group that is called DIFACIM. This is an acronym in Spanish that stands for Computer-Aided Drug Design. So what we do in our research group, it's basically basic research towards identifying bioactive molecules. So the mainstream is, of course, drug molecules, but also nutraceuticals, just small molecules that keep us healthy. So we use several computational approaches, mostly chemoinformatics, and on the right-hand side are listed the main research topics. The first one is actually one of the main topics of today. It deals with natural products and food chemicals. Also, regarding the target level, we are also working with epigenetic targets using this broad range of computational approaches. During the research, as many other groups, we developed some free resources, online tools that are available for everyone. And also, as part of the research projects, we, of course, use several databases that are already available. But for the natural product area, we're also, as I will mention later, building our own database that is also publicly available. So since we are at a university in Mexico, we are also interested with education and dissemination of science. And one of the kind of the latest contributions from several students in the group, it's to put together an electronic handbook for teaching. So this is meant for use to be used in chemoinformatics classes in Mexico, but it's also kind of thinking for self-studying. So now we know that most literature, it's in English. This is imperative, but also sometimes like students in Latin America wants to get started with their own language. So this particular electronic handbook is written in Spanish. And all the code is freely available. It's made on Colab. Okay, so let's move to some basic definitions that you are quite experts already, just for the sake of completeness. So let's start with the concept of chemical space. So in the literature, we will find several formal definitions of chemical space. Here is just a short list of those definitions. In particular, I'm highlighting here the one proposed by Professor Barnek that says that it is an ensemble of graphs of descriptor vectors that, as you can see, the objects, I mean, the compounds should be defined in some way, but some sort of descriptors. Along the same lines, the group of Raymond also says that chemical space is the group of all possible molecules described by the chemical properties. Yet another definition, it's listed, well, it's provided here at the top, by Bishop and colleagues in 2013, that we can basically translate this definition as a table, I would say. So in this table, we will have chemical compounds that can be from any nature. Of course, I think in the room, most of the talks have been dealing with small molecules, like for drug discovery, but I think that formally speaking, the chemical space can encompass everything. With chemicals, of course, on-demand compounds, but why not organometallic compounds, peptides, basically any chemical. Now, in the columns, we will have the descriptor or descriptor space. Now, for most practical applications, we know that the chemical space concept is used a lot along with the visualization. So if we have a multidimensional descriptor space, we know that we have to use some sort of dimension reduction technique, so we can project the high dimensional space into 2 or 3 dimensions. And so we can come up with kind of a scatter plots, 2D or 3D plots, or even networks that are being used a lot nowadays. Now, if we map a property, any property that we want, including, of course, biological activity, then we can use or we can perform a structured property relationships. And also, for the practical applications of the chemical space concept, as we have seen already, they are used a lot for structured property relationships, for the structural diversity analysis, library design, compound selections, and many other applications. So I'm not going to spend too much time on this, but you know that there are plenty of visualization methods to deal with these high-dimensional chemical spaces. Now, let's just make a pause and let's talk about a little bit of physics. Multiverse. So in physics, the concept of multiverse proposed by Everett says that it's a hypothetical collection of potentially observable universes, and each universe is defined by its own set of properties. Now, if we put together the concept of chemical space with multiverse in physics, I think we can suggest the concept of chemical multiverse. So the chemical multiverse would be the group of numerical vectors that describe the chemical space from the set of compounds, but in different manners. And each chemical space, as a multiverse, would be defined by its own set of properties. Just kind of for the sake of illustration, here we have like a cartoon representation of the chemical multiverse concept. Say that we have a given set of N compounds. If we wish, we can describe the chemical space of the set of N compounds using, let's say, continuous properties, drug-like properties, wherever you want. That would lead us to the chemical space that is in blue. But for our project, we want to use, let's say, fingerprints, max keys, extended connectivity fingerprints. So for the same set of compounds, we can define a yet different chemical space based on different descriptors. And we can keep going. For the same set of compounds, we can choose a different set of representations, let's say constitutional descriptors, and we will have a chemical space that is there at the bottom in pink. So each one would be a valid chemical space. And the question is, which one is better? And sometimes we don't know. It really depends on the goals of the project. So here the idea is quite simple. Instead of focusing on a given set of descriptors, why not using multiple descriptors, multiple chemical spaces, or why not analyzing the chemical multiverse, like the ensemble of those chemical spaces? So this is an illustration of the chemical multiverse using real compounds with real methods. Also for the illustration purposes, we have there chemical multiverse of 5 data sets from different sources. In the first column, we have typical drug-like compounds. Second column, we have compounds that interfere with protein-protein interactions. In the middle, peptides. Fourth column, natural products from Mexico. We'll talk about that later. And last column, food chemicals. So at the top, the visualizations, chemical space are done with a map4 fingerprint, just for illustration. And at the bottom, continuous properties. So in both cases, the chemical spaces are produced using PCA. So we can clearly see that depending on the descriptors that we're using, the relationships of the compounds between each other change or can change dramatically. Perhaps the most dramatic change is in the case of peptides. So in the middle here, according to the map4 fingerprint, all peptides kind of look alike, but if we change the description, we can clearly see a difference. So again, the idea here is for have a kind of comprehensive or complete view of the chemical space of these chemicals. We can use both representations. And of course, we can use much more than two if we want or if we need. Okay, now, also for kind of introductory purposes, let's talk about very briefly about natural products in drug development. So we know that natural products have been around and probably will be continuing around for so many years. So I took that picture from a nice review of Drs. Maria Valli and Bolzani, highlighting some key natural products throughout the history. So in the literature, there are plenty of reviews highlighting natural products that are used now in the clinic or that has inspired, as also Professor Schneider mentioned, has inspired the development of actual drugs. Here is just a brief overview of some examples of really important natural products. So in this slide, I'm showing contributions from natural products from Latin America, different countries in Latin America that are now in the market, that are in the clinic. So as in many other areas, artificial intelligence, computational methods have been playing a lot of roles in natural product drug discovery. Just for the extraction of natural products, the convolution, identify active compounds is used a lot. There's a wide range of computational techniques. And also as the other techniques that we have been hearing about, I've been using a lot to identify bioactive compounds from natural origin. So if we have a natural product database that is shown on the right-hand side, that database, as any other database, can be used as a starting point for chemical space analysis to develop natural product likeness scores, for the deorphanization, a source of inspiration for the novel design, as also Gilbert has already mentioned, and also Connor Colley. And of course, the prediction of bioactive functions based on those natural products. Of course, like the center stage are the databases. And also, you're quite familiar in this room, the high importance of molecular databases. Just very briefly, just again, just for the sake of introduction, we know that there are many databases of different sources, can be for general screening, focused libraries, de novo, on demand. But I would say, do not forget natural products and food chemical databases. So very recently, well, we have been working with food chemical databases for quite some time. This is probably the latest example, analyzing the contents and diversity of, I think, the largest public database of food chemicals is FoodDB, with more than 77,000 food chemicals. It's freely available. And here the basic question is, what's the diversity? What are the main molecular scaffolds, the distribution of properties? In this slide, I'm just showing illustration of the analysis of the chemical space based on two different representations. At the top, chemical space that, by the way, it's with Disney, was obtained using max keys, and at the bottom, extended connectivity fingerprints. Food chemicals, food EB, are in kind of that reddish color. Just for reference, approved drugs by the FDA are in orange. In green are food chemicals that are commercially available. And also, just as a reference, in yellow, it's represented a diverse set of natural products. So just comparing the chemical spaces based on the two different fingerprint representations, we can visually see that there is a nice overlap between food chemicals and approved drugs. And actually, if we kind of see what are the exact coincidences, you could be surprised that so many drugs, I mean, not so many, but several drugs that are approved by the FDA are food components, are food sources. Also, I think it's quite interesting that, as you can see kind of in the outer part of the both chemical space representations, food chemicals are really expanding the traditional chemical space of FDA-approved drugs. So I think kind of the take-home message for this type of analysis, again, we did several other analyses that you can check out in the paper, is that the food chemicals have really interesting features for finding bioactive components in a systematic manner. So now let's move to natural product databases, emphasizing here the contribution from Latin America. First, I'd like to highlight that Johannes Giermeier, a few years ago, did a very nice review of natural products in the public domain. As you may know, several years ago, the most comprehensive natural products are, and still are very important ones, commercially available, but several research groups around the world have been developing natural product databases that are in the public domain. And Johannes did a very nice review and analysis of the compounds in those databases. 2 years later, Christoph Steinbeck and Maria Sorkina start to building, you know, that famous database, Coconut, that is like putting together all those natural products in the public domain. So this is the, I'm sure you know the website of Coconut that now is under a major update. And I think later this year, the group of Christoph is going to make a new big update of Coconut. So in Latin America, I think one of the first groups, one of the first countries that they start putting together a really nice comprehensive database of natural products. It's in Brazil. Specifically, it's a cooperation between the São Paulo State University and the University of São Paulo. This is the current website interface. And at the bottom, it's listed the publication that goes back to 2013. So this is just a continuous part of the website where the user can find, filter the contents of the database. I think that the website is really nice design, and Nube is also part of the Coconut database, and it's also part of the Sync database, and can be also found in ChemSpider. So 2017, they made a new update, and I think also later this year or next year are gonna publish a next big update of Nube. So in cooperation with the group in Brazil, with Marilia, Adriano, and Vanderland Bolzani, we did like chemical space visualization, contents, diversity analysis of the compounds in that, I think, a very important collection. And Nube has been used in several publications, several studies for virtual screening. Just back there is an example where they actually find active compounds for T-Cruci using kind of a standard virtual screening methods. So more recently, 2023, Miguel Ángel Chávez from Peru and his colleagues put together the first Peruvian natural product database that is also publicly available. This is the website. They actually developed a very nice application for the smartphones. And in collaborations with Miguel Angel, we did again visualization of chemical space, profiling of the properties of that collection. And I should like to mention here that the first version of the Peruvian natural product database is not that big. It's 200 compounds, should be tiny, but as you know, all major databases start from small, and over the years, of course, are building up. I'm highlighting here the size because when we analyze the chemical space, the diversity of the Peruvian natural product database with other bigger databases from Latin America, including the Nubi one, Biofakim, that is from Mexico, mentioned in a minute, and also natural products from Africa, from AfroDB. We can see at the bottom of the slide that the Peruvian natural products in magenta are quite diverse, are covering as much as the space as any other databases. And again, this kind of makes us to think that the larger libraries are not necessarily the most diverse. Other countries like in Panama, El Salvador, Costa Rica, independently have been developing the sets of natural products on their own. In some cases, individually or collectively, have been actually used to identify bioactive compounds. Back there is just an example of that. So in Mexico, also now for the first version was published in 2019. We start putting together natural product database from compounds that are isolated and characterized in Mexico. We call database Biofakim, and in many research groups at UNAM and other universities work on this isolation, characterization, bioactive components. Traditionally, a lot of people rely on traditional medicine that works for them. And here the rationale is kind of why these compounds are so effective. So here the idea is kind of simple, just put everything together in a single repository so that everybody can kind of start making this structure property of function relationships and to do virtual screening, identify bioactive components. So the current version of this database has 605 compounds. Of course, most of the work, as you know, it's data curation, that the students don't like it that much, but they still are necessary. Hopefully they can hear the talk that it's important to curate data. And the most recent development is putting a website freely available to explore the contents of this database. That is the website, and the user, well, over here, there is like a link where it's possible to download the entire database. It has SMILES strings, has bioactive activity as annotated in the publications, and each compound has published reference. So if you identify any compound that is of interest of you, you can go to the original publications and even contact that research group that isolated the natural products, or you can contact us and we can be kind of a bridge between your group and the group in Mexico. So a couple of other databases also in Brazil. Marcos Scori recently published in JCIM a major update of the database that is called Sistema X. And Martina Vecchia in Argentina is also developing a database from that country. So since there are many like similar efforts but done independently, the idea was and still is, why not communicating? Why not putting together the efforts? And we kind of decided between different groups to build and unify and create a Latin American natural product database. So we're calling this database LANA-PDB, Latin American Natural Product Database. At the top are the different universities and institutions contributing to this collection. And the current version, as I will show briefly in a moment, contains the second version, roughly 13,000 unique curated compounds. So this is the team. We're going to put faces to the names. And here I'd like to really, really recognize the work of Alejandro Gomez is a PhD student in our lab, collecting all this information. Of course, each database has its own format. So what he's doing is putting together in the same format. It's a very labor-intensive job. And also it's a great opportunity for Alejandro to communicate directly and interact a lot with research groups in Latin America. So in William Zamora in Costa Rica, Dionisio Olmedo in Panama, as I mentioned before, Vanderland, Adriano, and Marilia Valli in Brazil, Miguel Angel in Peru, and Nector and Johnny in Colombia, and Marvin from El Salvador. So this is basically the team. So this is the contents, a little more of the contents of the database. There are listed some of the, actually there are 10 databases. We have 3 databases from Brazil, the one from Colombia, Costa Rica, and El Salvador. Each database has a different number of compounds. This is a continuation of that list. In addition to Biofakim, another institute at UNAM, Instituto de Química or Chemistry Institute, has been also developing their own database. We're collecting that information, and the database from Panama and from Peru. Now, each database it's been updated independently. So what Alejandro is, in addition to put together that information, is when the other database from Latin America is being updated, he updates the Latin American natural product database. So that's a lot of communication and a lot of back and forth process. All the details of the first and the second version are listed there in the publications at the bottom. So, of course, in addition to constructing and analyzing the database, other parties analyzing the contents. So this is a visualization of chemical space of Lana PDB using 2 types of fingerprints, max keys and map4. So each database from different countries is color-coded by a different color. The ones in Brazil, in both cases, are in yellow, in Mexico are in green, and so on. So in addition to the visualization, of course, we are doing the distributions of the fingerprint-based similarity, property similarity, and maybe not surprising, the Brazilian database is the most diverse, followed by the one from Mexico. But again, Peruvian natural products, surprisingly, again, despite the small size, the chemical structures are quite diverse and different from natural products from other sources from Latin America. Okay, so the short version here, the short part. So why? As you have seen, we have been analyzing chemical spaces of several different databases, and we were starting to think, why not looking at the same pictures in a different manner? So we kind of got inspired by the quote from Robert Henry here that says that the object isn't to make art, because we are chemoinformaticians, but it's to be in that wonderful state which makes art inevitable. So, as I said, we have been working with food chemicals, with epigenetic targets. There are several food chemicals that play a major role in our normal everyday epigenetic processes. So this is like a very formal chemoinformatic analysis of food chemicals, and a new database that we developed that is called EpiFood. EpiFood are known food components that, from the experiments, we know that they act as modulators of epigenetic targets. So based on that figure, we can do structure property relationships and so on and so forth. But talking with a student that made this chemical space analysis, we were wondering, why not just taking out the axis, take out the chemoinformatics part, and you may end up with a nice figure that probably anyone outside the university may like it. So we kind of started thinking about making like nice, perhaps artistic views based on the chemical space concept. So very briefly, so I'm going to show examples of artworks that have developed some students as part of their normal projects. These figures are examples done by Daniela Gaitan and Ana Chavez, analyzing food chemicals. So all figures are done with D-maps that we can call like technique. So the descriptors in this case are common fingerprints, and the compounds there are color-coded by the flavor category. So in addition to diversity, structure property relationships, we think these figures for some people may just look nice. This is another example done by Edgar Lopez. He is analyzing natural products. Actually, in that figure, we have 1,000 natural product subset. So this figure was done with Data Warrior using self-organizing maps. So again, as part of the Edgar project is to study structure property relationships. But again, I mean, for Edgar and for so many others, they look like a horse. Can be interesting, what kind of look like. And I think this type of visualizations can help us to communicate our chemoinformatics, really focused intensive science with just people outside the walls of the university. So that's the last slide in that regard. So we're putting together what we're calling a chemical art gallery. So if the students came up with a nice figure that they like it, they can send us that picture and deposit this picture in this chemical art gallery. So if you came up with a nice visualization for your projects, feel free to include that figure in the chemical art gallery. So in summary, so we have talked about the importance of chemical space, chemical multiverse, as a conceptual basis for molecular database analysis. Also kind of talk very briefly natural product databases, emphasizing here the Latin American natural product database. Currently, we have 10 databases with around 13 compounds, and all compounds, all the information can be freely is freely accessible in that website. So I want to thank again all of you for your attention, like students. This is kind of brief timeline, different years. So this is the most recent picture that we took a few weeks ago that Bill Jorgensen visited us for a conference. And then you are all invited to visit our Mexico City, our campus. This is the School of Chemistry. It's a large university. So overall has like 5 buildings. This is the main one. And there are like 7,000 students in the campus. The concert hall, soccer stadium, was built in 1968 for Olympics in Mexico City. The main library, and that's beautiful building of the School of Medicine that is just next to the School of Chemistry. My last slide, and advertising. So you're all invited to submit the papers to a special issue of Molecular Informatics that is focused on the exploration of big and ultra big chemical spaces. So thank you so much for your attention.
The Latin American this Latin American database very interesting. Is it just an electronic database, or are the compounds have been plated? Are they available for experimental screening?
Yeah, actually, yeah, that's a good question. Most of those compounds are physically available for screening. So also part of that database, the annotations, it's the commercial availability. So they can directly be purchased.
Or they're just powders?
Mostly powders are not plated. So, but yeah.
And centralized in one place, or you need to ask your partners to
Yeah, at the moment, you need to contact every partner. So kind of in the near future would be the idea now to centralize and physically available the same, the large collection. Yep.
Right, thank you very much.
Thank you.